This audio lecture is about antibiotics. An antibiotic is a substance naturally produced by a few bacterial and fungal species that inhibits or kills bacteria. Let's talk a little bit about how we used to treat people that had infectious diseases before we had antibiotics. We used to do things like give them enemas, put them into blood-curdling ice baths, because often when people get infectious diseases, they get fevers. So they used to put them into blood-curdling ice baths, blood-curdling because of the screams from the people getting put into the terrible ice baths. Deadly starvation. I think people sort of knew that there was something inside these people that was making them sick, so they wanted to starve whatever was inside of them. But unfortunately, they often starved the actual people to death as well. Even when George Washington was president of the United States and he had access to the best medical care, he actually had a really bad sore throat. His throat was really red and there was pus in the back of it. And the best doctors in the world did the best treatment at the time, which was bloodletting. And they actually removed 2 liters of blood from him in 24 hours. So just a little factoid, not just physicians used to do bloodletting, but barbers did. So if you just cut hair, your barber pole just had a blue stripe on it. And if you were a barber that cut hair and did bloodletting, it had a blue stripe and a red stripe to signify that you did bloodletting as well. All of these things, by the way, would actually decrease your body's defenses and actually aid in the infectious disease or aid in the illness. We didn't have the germ theory of disease until the late 1800s, but even then, little could be done for people with infectious diseases. 100 years ago in the United States, a third of children were expected to die of infectious disease before the age of 5. So let's talk a little bit about the history of antibiotics and antimicrobial drugs. We mentioned back in the introductory lecture Paul Ehrlich. He was a German chemist back in the early 1800s, and after the germ theory of disease came out, Paul Ehrlich was like, if there's microscopic organisms that can make people sick, then I want to invent a magic bullet, something that will kill these microscopic organisms but not harm the host, in this case humans. So he literally synthesized hundreds and hundreds of compounds. All of these compounds contained arsenic and phenol. Phenol because of Lister, and arsenic because at the time people sometimes killed each other slowly by adding arsenic to each other's foods. And right before they died of the arsenic poisoning, if they had an infectious disease, the infectious disease had gotten better. Compound 606, so the 606th thing that he synthesized that he called salvarsan, was actually given to patients in 1910 to treat the infectious disease syphilis, so the sexually transmitted disease syphilis, which was rampant at the time. It was only used for a few years because it was actually not selective enough. It was actually quite toxic because of the arsenic and the phenol. Salvarsan is not an antibiotic because it was synthesized. It's actually considered an antimicrobial drug. Another interesting person is Gerhard Domach. In 1932, he tested a red dye called Prontosil for antibacterial properties. In animals, it inhibited Staphylococcus bacteria and Streptococcus bacteria and some other gram-positive bacteria as well. And his daughter was learning how to sew from his wife, and she accidentally pricked her finger with the sewing needle, and she got a really bad infection. So bad that they told him the only thing that could be done was for her arm to be amputated, because now we would know she actually had a Staphylococcus infection. Obviously, they did not want to do that. So he told his wife, listen, we are testing this red dye called Prontosil, and it's working in animals. So in 1935, he injected Prontosil into his daughter, and she slowly actually got better. She gradually improved, and in 1939, he won the Nobel Prize. Prontosil would not be an antibiotic. It would be an antimicrobial drug again because it is a red dye. It is not something that's produced by a few fungal or bacterial species. Then there's Alexander Fleming, In 1928, he was doing research on Staphylococcus bacteria, and he was leaving in a hurry, and he did a bad streak plate. He kept the lid on his streak plate too high, and Penicillium mold spores, microscopic spores from the air, got into his streak plate and contaminated it. But when he came back after the weekend, and he sort of noticed that, hey, where bacteria should be growing, it isn't, because this penicillium, what he identified as penicillium mold, is growing. So he hypothesized that the penicillium mold was producing a substance that was actually killing the bacteria. And he named the substance that the penicillium mold was producing that was killing the Staphylococcus bacteria penicillin. He actually tried for years to isolate the substance, and he couldn't do it. But he did write a paper, and two chemists named Howard Florey and Ernst Chain read the paper and decided they would try to isolate the substance. And in 1942, they actually produced enough of penicillin for the drug to be available for human use. And in 1945, Fleming, Chain, and Florey actually all shared a Nobel Prize. Now you can imagine that when people realized that a mold, penicillium, produced a substance called penicillin that could, and penicillin is an antibiotic because it's produced by a fungus, a mold, penicillin, that when there was something, in this case a mold, that could produce a substance that could kill bacteria, people were going to go looking for more. And a microbiologist named Selman Waxman was the perfect person to do this. He actually was a microbiologist that studied microbes, bacteria that live in soil. So in 1944, he isolated streptomycin, an antibiotic, from bacteria called streptomyces that live in soil. And he went on to discover tetracycline, erythromycin, 20 different antibiotics total. And he was the first person to coin the term or use the term antibiotic. He said it was anti for against and biosis for life. And in 1952, he was awarded a Nobel Prize. So there are several different, as I mentioned, genus of bacteria and fungi, mold in particular, that can produce antibiotics. Streptomyces, that genus of bacteria, produces about half the antibiotics that we have discovered. Antibiotics including erythromycin, streptomycin, tetracycline, chloramphenicol. The genus Bacillus, which is a bacteria, produces bacitracin and polymyxin B. The mold Penicillium produces penicillin. The mold Cephalosporin produces the antibiotic cephalosporin. So it's just a couple genus of bacteria and molds that produce antibiotics. We're not sure why microbes actually produce antibiotics. We haven't found them in nature, the antibiotics, in great enough concentrations to actually inhibit or kill each other. So it isn't some sort of like intermicrobial warfare. And it's very, very time consuming. There's lots of steps for a bacterium or mold to produce antibiotics, and it takes a lot of energy. It takes a lot of ATP. It takes a lot of their actual like macromolecules and nutrients to do it. So we actually don't know why they do it. Now, if you asked a microbiologist like me, what would be the ideal qualities of an antibiotic or antimicrobial drug? This is my dream antibiotic. I would like it to inhibit or kill the growth of a pathogen, but cause no damage to the host. I am all in on Ehrlich's magic bullet idea. I would like it to cause no allergic reaction to the host. About 0.1% of people are allergic to penicillin. I would like it to be stable as a liquid or a solid. I'd like to be able to prescribe it as a pill, to give it as an injection, or to give it as an IV. I'd like it to remain in specific tissues long enough to be effective. and not be broken down by the liver, for example, too quickly so that it isn't going to be effective. I'd like it to inhibit or kill pathogens before they become resistant to it. We're going to talk about resistance in a moment. I'd like it to be inexpensive, and I'd like it to not kill any of our normal flora. Now, notice in this I said inhibit or kill a few times. That's because we do have an amazing immune system, which we're going to talk about in several of our future audio lectures. Now, this ideal antibiotic or antimicrobial drug doesn't exist, but hey, we all have to dream. Now, once they discovered these antibiotics, the first thing they did was set out to figure out how they actually kill bacteria. So we have these amazing antibiotics. How are they doing what they do? So they went out to find the targets of antibiotics and antimicrobial drugs. And here they are. The first target of antibiotics or antimicrobial drugs is bacterial cell wall synthesis. How they do this is they block the synthesis of peptidoglycan. We talked about this back in prokaryotic cell structure. Gram-positive bacteria have a thick cell wall. Gram-negative bacteria have a thin cell wall. But basically what this does is it can alter their osmotic pressure. But when you think about this, and when we think about these targets, this wouldn't work for both gram-positive and gram-negative bacteria, because if we think back to their cell envelope, gram-positive bacteria have two layers to their cell envelope. A thick cell wall made of peptidoglycan and teichoic acids, and underneath it, the cell basic that is a cell membrane. Gram-negative bacteria have a cell envelope made of an outer membrane made of LPS, underneath that, a skinny little cell wall made of peptidoglycan, and underneath that, a cell basic that is a cell membrane. Antibiotics that target the cell wall, block the synthesis of peptidoglycan, would only actually be effective against gram-positive bacteria because the outer membrane actually blocks access to the cell wall in gram-negative bacteria. So in gram-positive bacteria, they actually would die from these antibiotics because of the osmotic pressure issue. If you actually target their cell wall, that's the only thing they have protecting them. And depending on the environment, they would shrivel or they would burst, and that's how they would perish. Antibiotics that are examples here are something like penicillin, vancomycin, or amoxicillin. Our second target is disrupting membranes. I just said membranes here, so this could be a cell membrane or an outer membrane. They will disorganize the structure of the membrane, or they will inhibit the function of the membrane. An example here would be the antibiotic polymyxins. Now, think about our cell envelope structure again. These would only be effective against gram-negative bacteria because they have the outer membrane made of LPS. It would work against that and leave their skinny little cell wall exposed, which is really tiny, and then they'd have their tiny little cell membrane right underneath that. Cell wall is not going to be hard to get through here. Now, the gram-positive bacteria wouldn't be effective against because they have that big, thick gram-positive cell wall. It's not going to be able to get through that 300 layers of peptidoglycan. That gram-positive thick cell wall is protecting their cell membrane underneath it. Now, selectivity is an issue here because prokaryotes and eukaryotes both have the cell basic cell membrane. So we actually don't use these systemically because it would be toxic to our actual cells. So these are only given topically. Third target is nucleic acid synthesis. We can block the synthesis of DNA or RNA. These don't seem like on the surface that they would be a good choice. First of all, let me say that these are going to be effective against both gram-positive and gram-negative bacteria. These antibiotics are very small. They can get past in gram-positive bacteria, the thick cell wall. In gram-negative bacteria, the outer membrane and their thin cell wall. Now, you would think that this would be a selectivity problem again, because after all, we have nucleic acids, but they actually do work. Let me give you two examples. There's an antibiotic called rifamycin. It targets bacterial RNA polymerase. Our eukaryotic RNA polymerase is different than enough from bacterial RNA polymerase. And then an antibiotic called quinolones, it targets bacterial DNA gyrase. Our eukaryotic DNA gyrase is different enough, so these work just great. Our fourth target is blocking protein synthesis. Prokaryotes have 70S ribosome. Eukaryotes have 80S ribosomes. So the difference in the size of the ribosomes here is going to make these a perfectly great antibiotic target. Again, these are going to work for both gram-positive and gram-negative bacteria because these antibiotics are small enough to get past the outer membrane, skinny cell wall of gram-negative bacteria and thick cell wall of gram-positive bacteria. Antibiotic examples here include erythromycin, streptomycin, and tetracycline. So again, effective against both gram-positive and gram-negative bacteria. And then our last target is metabolic pathways. Here, the antibiotic is going to work against, or not antibiotic, these are going to be antimicrobial drugs actually. They're going to work as competitive inhibitors and occupy the active site of the enzyme, so the true substrate cannot bind. What they're really going to do is inhibit metabolism, usually folic acid metabolism. So organisms need folic acid to produce DNA and RNA. Eukaryotes like us, we get folic acid from our diet, from the food that we eat. We don't produce it by ourselves. But prokaryotes, bacteria, they have to produce their own folic acid. So if you actually use an antimicrobial drug like the sulfa drugs, Bactrim is a great example, you block their ability to actually produce folic acid, which means they can't produce DNA and they can't produce RNA. That means they can't make any more bacteria because they have to make a copy of their DNA to do binary fission to produce more of themselves, and they can't do things like transcription and translation. Antimicrobial drugs that target metabolic pathways would be effective against both gram-positive and gram-negative bacteria. They can get past the cell envelopes. Now, I want to say there is a tiny teach just on the antibacterial and antimicrobial drug targets. So if you want to hear me just go through these again, you can listen to that tiny teach. I also want to say that viruses have none of these targets. So antibiotics are only appropriate for bacterial infections. Antibiotics are not appropriate for viral infections. Viral infections include the common cold. They include the flu and other viral infections, which we're going to talk about here in a few minutes as well. Now, there is a distinction I want to make between a narrow spectrum antibiotic and a broad spectrum antibiotic. A narrow spectrum antibiotic or antimicrobial drug is only effective against gram-positive or gram-negative bacteria. To use this, you have to know what the pathogen is. So for example, you did a gram stain and you see that you have gram-positive bacteria. So you can use a narrow spectrum antibiotic. So for example, this is using penicillin that targets peptidoglycan synthesis. So you know that that's only effective for gram-positive bacteria. So that would be a good choice. A broad spectrum antibiotic or antimicrobial drug is effective against both gram-positive and gram-negative bacteria. This is what you use when the pathogen is unknown. You didn't do a gram stain. You don't know whether it's a gram-positive or a gram-negative bacteria. This would be using, for example, an antibiotic like tetracycline that's effective against both gram-positive and gram-negative bacteria because it targets the 70S ribosome. Broad spectrum antibiotics have more side effects. They kill more normal flora because when you take an antibiotic, it's not just going to kill the bacteria that's making you sick, it's going to kill normal flora as well. And in this case, it's going to kill both gram-positive and gram-negative normal flora. They also put you at risk for a secondary infection. This is because it's killing normal flora, it's going to open up some valuable real estate that another bacteria might take up that valuable real estate. That would be a secondary infection. It also puts you at risk for a super infection. This is something like Clostridium difficile. So Clostridium difficile should be a very small percentage of the bacteria in your gut. But if you take broad spectrum antibiotic after broad spectrum antibiotic, well, Clostridium difficile is genus Clostridium. So when you take antibiotics, they will produce endospores because the endospores can survive the antibiotics. What you can end up with in your gut is basically just Clostridium difficile endospores. When the antibiotics end, then the Clostridium difficile endospores will become vegetative cells or bacteria, and that's all that will remain in your gut. That is a super infection. And then those bacteria will do things like produce exotoxins and extracellular enzymes that we're going to talk about in the infectious disease audio lecture. You can do things like take probiotics. Probiotics to, for example, give an antibiotic something else to kill besides your normal flora. Sort of if there's going to be collateral damage, let that collateral damage be to probiotic bacteria. Just make sure that you take the probiotic 2 hours apart from your antibiotic. Now, how can we determine which antibiotic or antimicrobial drug to use instead of just sort of guessing? Well, we can use what's called diffusion susceptibility testing, or what's also known as the Kirby-Bauer test. You guys actually do this in your labs that you do. What we do is we inoculate Mueller-Hinton agar with the sample of the bacteria. Mueller-Hinton agar is used because it's a very neutral media. We want the bacteria to behave the way they would in the human body. So it's very neutral. And then we arrange discs that are impregnated with different antibiotics. And we look to see if the bacteria did not grow around the antibiotic discs. If they do not grow, that means they are inhibited by the antibiotic in that disc. The larger the zone of not growing, the better. So that way we're not just guessing. These this disc diffusion susceptibility testing is very important if a patient has acquired the infection in a hospital or healthcare setting, because that is likely going to be multiple antibiotic resistant. Now, how do we pick the route of administration? How are we going to give you that antibiotic? If it's not anything very severe, we might just use a topical application. Apply it directly with something like a cream. The most common is to give you the antibiotic orally with a pill. That's great because the patient can self-administer it. We do use a lower dosage, so we need a longer dose time. But we do worry, will the patient continue the entire course of the antibiotic? This is very important because stopping short, saying, well, they gave me a prescription for 7 days or 10 days, but I feel better after 3 days, so I'm just going to stop, can actually increase the possibility of the bacteria becoming resistant to that antibiotic. Or we can use something like an injection or an IV. Now we can use a higher concentration, so we can do it for a shorter period of time, but of course needles are involved, which some people don't like, and a healthcare provider has to be present, either to give the injection or to administer something like the IV. Now let's talk about resistance to antibiotics and antimicrobial drugs. The good news is few bacteria are naturally resistant. The bad news is bacteria can acquire resistance by mutation, conjugation, transduction, and transformation. We're going to talk about mutation, conjugation, transduction, and transformation when we talk about microbial genetics. So not here, but in microbial genetics. Now I want to talk about how bacteria can resist antibiotics and antimicrobial drugs. So how they can do that. There is a tiny teach on this, so you can listen to that in more detail, but I'm going to really briefly talk about it here. So here are the ways that bacteria can resist antibiotics. The first way they can do it is they can produce enzymes that destroy the antibiotic. So for example, bacteria can produce an enzyme called beta-lactamase that actually destroys antibiotics like penicillin. It literally breaks the penicillin antibiotic apart. They can decrease entry of the antibiotic into the bacterium. So they can do this, for example, by altering the permeability of the cell membrane to exclude the antibiotic. So the antibiotic can't get into the bacterium. They can pump the antibiotic out of the bacterium before it can act. These pumps are embedded into the bacterial membrane, and these pumps are capable of pumping any antibiotic out of the bacterium. They can alter the target site of the antibiotic. So for example, they could alter their peptidoglycan of their cell wall slightly. They could alter their DNA gyrase slightly, so the antibiotic cannot alter or cannot bind to their target. They could change their metabolic pathway. So for example, the bacteria can now use folic acid in the environment and no longer needs to produce folic acid. So an antibiotic that targeted the synthesis of folic acid would no longer be effective. Now I want to spend just a few minutes talking about antibiotic resistance and a few examples. First example I want to talk about is MRSA, which is probably the antibiotic resistant bacteria that everybody's most familiar with. MRSA stands for methicillin resistant Staphylococcus aureus. It doesn't mean that this bacteria is just resistant to the antibiotic methicillin. It's actually resistant to methicillin, penicillin, amoxicillin, oxacillin, and many other antibiotics as well. About 33%, or 1 in 3 people, carry Staphylococcus aureus on their skin and in their noses. Everybody carries Staphylococcus epidermidis on their skin as well. About 1 to 2% of the 1 in 3 people who carry Staphylococcus aureus on their skin and in their noses. So let me redo the statistics because that sounded confusing. 33%, or 1 in 3 people, carry Staphylococcus aureus on their skin and in their nose. 1 to 2% of those people, or about 2 in 100 people that carry Staphylococcus aureus, have multiple antibiotic resistant Staphylococcus aureus, or MRSA. The US and Japan have the highest rates of MRSA infections. MRSA is an example of a superbug, meaning it's resistant to multiple antibiotics and antimicrobial drugs. Here are some other examples of superbugs. There's MRSA, which is multiple antibiotic resistant Staphylococcus epidermidis. This is the bacteria that covers all of us, our skin from head to toe. Mycobacterium tuberculosis is an example of a superbug. Pseudomonas aeruginosa, which is a healthcare acquired infection that burn patients get, cystic fibrosis patients get, sometimes people that have been on ventilators get. Salmonella is an example of a superbug. Neisseria gonorrhoeae, the sexually transmitted disease bacteria, is an example of a superbug. Resistance comes from one of the things that resistance comes from is improper use of antibiotics. Antibiotics and antimicrobial drugs should only be used for bacterial infections, never for viral infections. So for example, 50% of antibiotic prescriptions written for sore throats are improper. If you have strep throat, that is a bacterial infection, so it is appropriate for you to get an antibiotic prescription. Any other prescriptions for antibiotics for a sore throat is inappropriate. 30% of antibiotic prescriptions for ear infections are inappropriate because the rest of ear infections are caused by viral infections. 100% of antibiotic prescriptions for the common cold are inappropriate. The common cold is always caused by a virus, so you should not be getting an antibiotic prescription. We need to educate medical doctors so that they are not inappropriately prescribing antibiotics. We need to educate patients so they understand that antibiotics are appropriate for bacterial infections but are inappropriate for viral infections. And if you look up at the screen, there's a campaign that the CDC started called the Get Smart campaign that shows patients these are caused by viral infections. These kinds of illnesses are caused by viral infections and should not be getting an actual antibiotic prescription. We have to remind patients that when you get an antibiotic prescription, we need you to take all of the medication as prescribed. We need to think about large doses before surgery. Why are we putting on people on like a broad spectrum antibiotic right before we cut them open and depleting their normal flora? Why after surgery are we putting you on another broad spectrum antibiotic? Can we wait and see if you get an infection again before we deplete all your normal flora? We need to use low dose, narrow spectrum if possible. Can we figure out what pathogen you have? Can we do things like a gram stain and the Kirby-Bauer test to figure out what antibiotic to actually use? And then a MIC, a minimal inhibitory concentration test, so we can use the lowest dose possible. Hand hygiene to decrease transmission of infectious disease. We need to do hand washing with soap and water, not hand sanitizer. And 80% of all antibiotics in the United States are given to livestock. And that is given to livestock to make them fatter faster and so they can live in less hygienic conditions. We need to stop that so that we humans don't develop resistance to antibiotics from only being exposed to them through our actual food chain. That is the end of this audio lecture. I'll see you on the next one.